Hello students, so welcome back to the channel. In this video we are going to discuss about the phloem, which is a type of a complex tissue. The phloem term was given by Nagle, and the function of the phloem is the conduction of food. It will help in the conduction of the food. So it is responsible to conduct the food from the leaves to the other part of the plant. Is it clear? Now, classification of the phloem on the basis of origin, there are of two types. Primary phloem, and primary phloem is the one which is developing from procambium. It developed from procambium. And development from procambium is occurring at the time of during vascularization. Means when the xylem and phloem formation is going on. Is it clear? And next is your secondary phloem. Secondary phloem development is occurring from vascular cambium. And it is occurring during the secondary growth, at the time of secondary growth. So depending upon the origin, two types of phloems are there, primary phloem and secondary phloem. Is it clear? Now on the basis of development, primary phloem is of two types. Protophlloem. Protophlloem means it is first formed phloem. As it is first formed phloem, it will have narrow seed tube. Okay, it is first formed phloem and it has narrow seed tube. But later formed phloem is known as metaphloem. You have to just remember it like a xylem only. Okay, so this is later formed, means afterward, later formed phloem. And as it is later formed phloem, we will say it has wide seed tubes, means bigger seed tubes, you can say. So on the basis of development, your primary phloem is of two categories. There is no two categories for secondary phloem. Is it clear? I hope up till here topic is clear. Next classification of phloem is on the basis of position. Is it external phloem? External phloem, which is normally present outside the xylem. Okay, it means it is present outside xylem. And when it is present outside xylem, you can remember example is generally your most of the angiosperms and gymnosperm. The most of angiosperms and gymnosperms, they are the one which are having this type of xylem. Sorry, this type of phloem. Okay, the one is your external phloem. Now second is your, this is now important, this is your internal phloem. Internal phloem is of, see one thing here, it can be also known as intraxillary phloem. Intraxillary phloem. Clear? Actually, it originates from procambium. It is also primary phloem, but it is present inner to the primary xylem, just like in bicollateral vascular bundle. Is it clear? So what is it? It is developing from procambium. It developed from procambium. It is actually primary phloem only. But one thing we have to remember, it is present inside the primary xylem. Inside the primary xylem. An example, bicollateral vascular bundle. Bicollateral vascular bundle. So let me just explain you. See here, See one thing, this is your, let us assume this is, I'm drawing it, check carefully the diagram. This I'm drawing the bundle 1, in which this side is the phloem present. This is outer side, let me label it, this is outer area, outer side, and this is inner side, okay beta? So phloem is present on the outer side, phloem is present on the inner side, and this is your xylem part. This is xylem, and this is your phloem, okay? So this phloem is present on the outside, but this phloem is present on the inside. So that is why you are calling this as an external phloem. This is external phloem, and this is known as internal phloem. So internal phloem will be present in the bicollateral vascular bundle. And see here, check the diagram. If I'm drawing it this way, check here. You should check here, please. This is phloem drawn only on one side. And this is your xylem present. So it means phloem is present, which one only? External phloem. So if phloem is present only on one side, then such type of vascular bundles are known as collateral vascular. So it is known as collateral vascular bundle. And if suppose the phloem is present both side as external as well as internal, then we say bicollateral vascular bundle. Is it clear? Now let me give you the examples of the bicollateral bundle families so that it will be easy for your learning. See here, the families are your Solanaceae, Cucurbitaceae, Convolvulaceae, Apocynaceae, and Excipitaceae. Now, third type of category is your included or interxylary phloem. Let me just draw it. Now, see very carefully, please try to focus it. About, if you check upside, check my cursor where it is, I'm putting it. Earlier, I wrote it intraxylary, okay? Now, this is interxylary. Means it is present within the xylem. So, how it is possible? Actually, it originates from cambium. It originates from cambium, vascular cambium generally, and it is secondary phloem. It is not primary phloem. It is secondary phloem. And they are present within the secondary xylem. And they are present within the secondary xylem. That is why we say it is within secondary xylem. Clear, beta? Now examples. Examples here you have to remember. Few families, plants are there. One is your amaranthus, Boravia, Chenopodium, and Salvadora. So there are three types of phloem depending upon position. Is it clear? Just wait, let me make it small for you so that you can get an entire picture also. So see here, on the basis of position, you are having three types: external phloem, your internal phloem, and included phloem. Is it clear? I hope up till here topic is clear to you. Now next we should know components of the phloem. So they are made up of four types of cells: sieve tube element, companion cells, phloem fiber, and phloem parenchyma. So just let me write first parenchyma, phloem parenchyma, and then phloem fiber. These three are living component and only one this is dead. Remember in the xylem, if you remember in the xylem, I'll just write it here, just for your learning sake, remember it. Xylem has three dead elements. Which three dead elements? Tracheids, vessels, and xylem fiber, and only one living element, that was xylem parenchyma. Is it clear? But in case of the phloem, phloem is having three living and only one dead. So these are the components of the phloem, and these components, check carefully, these are present in angiosperms. It means in the gymnosperms and pteridophyte, the component will change. So see here, in case of pteridophyte and gymnosperms, instead of seed tube, they are having seed cell. And instead of companion cells, they are having albuminous cells. Remaining will be same. They'll be having phloem parenchyma and they'll be having phloem fiber. Only difference is with respect to these two. They are having here seed cells and albuminous cells. Okay, but here they are having seed tube and companion cells. So now let us study one by one. Let's try to understand. Let us assume this is one cell, okay? This nucleus, and when it is dividing into two, the two cells are formed. This one cell and this one cell. This becomes as a sieve cell and this becomes as an albuminous cell. And this condition is seen in the pteridophyte and gymnosperms. But see here, check the diagram carefully. This is one cell. When it divides to form two cells, only one cell retain the cytoplasm only the one cell is having only a little bit cytoplasm. Another cell will have nucleus plus cytoplasm both. Nucleus is also there and cytoplasm. So this is nucleus present, okay? So this cell is known as seed tube element. And this is known as companion cell. So I hope you can understand from the diagram clearly that seed tube and companion cells are sister cells. Okay, they are sister cell. Now let me draw once again, check the diagram, I'll draw it here. This is your one seed tube cell and this is your companion cell, okay? So in between them, you are finding here, in between them, you will be finding here, check my diagram carefully. This is nucleus, okay? And this labeling I will do it as pit field connection. Pit field connection. It is very, very, very important line of NCERT because pit word we studied only in the sclerenchyma fiber or sclerids or in the xylem tracheids and vessels. But there we use word only pits and here I am saying it is pit field. Reason is that both the cells are living. They are living why? Because they are having the cytoplasm. They are having cytoplasm. But if you check my diagram, nucleus is present only in the companion cell. This nucleus is present only in the companion cell. So seed tube is the one which is living without nucleus. Seed tube is the one which is living without nucleus. I hope this much you are able to understand. Okay, but if you see here, check the diagram carefully, sieve cell and albuminous cell, they both are not sister cell. Is it clear? Though they are coming from same, but they are not sister because they are, see one thing, they can come from separately also. It can divide to form two albuminous cell. So it can divide to form two albuminous cell also. So it is not necessary that one cell has to divide to form sieve cell and albuminous cell. Albuminous cell can come separately from another cell. But here, if you see carefully, companion cell has to come from the same mother cell from where the sieve tube is coming. So in this case, they both are sister cell. Is it clear? Now let me explain you how the evolutionary sieve cell is becoming into the sieve tube, how the evolutionary it is going on. Now see, try to understand, this is one sieve cell, okay? This is sieve cell. It is little bit long and narrow, and see here they are having sieve pore. I'll not say these are the pits, okay? These are not pits, these are the sieve pores which are present on the lateral wall. These are not pits, please remember these are the sieve pore. I'm writing it here. These are sieve pore, but they are not present on the end, they are present on the lateral wall. Is it clear? They are the living cells, but they are having sieve pore on the lateral wall. Now check here carefully what we will do here. This is the one sieve cell, and I will place another sieve cell on top of it. Let us assume I am placing another sieve cell. Evolutionary, I'm trying to explain it. Then what will happen? They will try to dissolve the common wall. They will try to dissolve the common wall. Are you understanding the diagram? They're trying to dissolve the common wall. So when they're dissolving the common wall, let me draw it once again here. So this is the way how they are making the sieve pore. And these sieve pore are coming in the form of a plate. So it is known as sieve plate. And check carefully, I'm drawing it oblique. So they are having oblique perforation septa, which is now working as sieve plate. So this area is known as sieve plate. We call this area as sieve plate, and we say sieve plate has sieve pore. Is it clear? So it is, if you see carefully, it is like a tube-like structure, and they are longitudinally arranged. And on the side with every one, you can just, it's not with every one, but actually I'm drawing it here. These are the companion cells. Okay, these are the companion cells with nucleus present. And I'm using another color, check carefully. This is the common pit field connection. Pit field connection is present between the sieve tube and companion cell. So now I hope with this diagram you are able to understand that sieve cell is individual cell. It is individual cell. But sieve tube is actually syncyte. We will say it is a living syncyte. The sieve tube is living syncyte. Syncyte means fusion of many cells, and these cells are living only. All are living and they are becoming as a tube. That is why we say sieve tube. Understood? So sieve tube is a living cell without nucleus. Very important. Let me write here for you. So sieve tube is living cell without nucleus. This is the only cell in the plant kingdom which is living, but it is living without nucleus. Why we are saying living? Because it is having peripheral protoplasm. Is it clear? And all the functions of sieve tubes is controlled by the nucleus of companion cells. This much is clear. Now, additional information with respect to the competitive point, let me give it to you. See here, this is an additional information with respect to the competitive point. Check the board carefully. In the sieve pore, this sieve pore, callus deposition will occur. This is a callus deposition is occurring. Okay. On the sieve pore area, you are finding callus deposition is occurring. This is callus. And callus is nothing, it is a polymer of beta 1,3-glucan. It is clear. It is a polymer of beta 1,3-glucan, and this deposition will take place during the autumn season, means when the leaf has to fall down. So they will slowly clog it like this way. So total clogging is going on, As a result, leaf fall will be there. Are you getting the idea? This is the way how this deposition is responsible for the leaf fall. So you can say it will be seen in which season? It will be seen in the leaf fall seasons, or you call it as autumn season. In these times they are going to form. And this is structure what they are making like this way. We call it as either you say callus plug or either you say it is callus pad. Different names are given. In the competitive questions, you will be finding this language. It is there. We call it as either callus pad, okay, or either callus pad. This is the term they are using it. And it is made by those plants which shed their leaf. Those plants which shed their leaf are also known as deciduous plant, not evergreen. Pinus is evergreen, okay? Pinus tree, which we studied in the gymnosperm, is evergreen, but the deciduous one, they will be the one which are shedding their leaves, so it will be shown by the deciduous plants. Another is this, it is actually this pad, callus pad, what they are making it. It is useful in preventing the spreading of the infection of fungus. So you can say another role it is, it protects sieve plate from bacterial infection or spreading of the fungus infection also, bacterial or fungal infection. Sometimes what happens, this another element is also helping in making the callus plug. Check my diagram carefully. Sometimes these are the proteins which are also present in between them, which are helping them to make the callus pad. So these proteins, please focus, these proteins are your P protein. P protein, P means phloem. Present in the phloem known as P protein. So they are also helping in function in making the callus, or you can say they are helping in the wounding, also healing of the wound. And you can understand simple example taking a kitchen sink example. Okay, in the kitchen sink, if you know, in the center one is there, structure is there which is having pores. So imagine that structure is like your sieve plate, and the pores are nothing sieve pore. Okay, and now if suppose your mother is washing the vegetables and small pieces of vegetable get stuck, then water movement will be slowed. The same way, just imagine the small vegetable getting plugged, getting trapped there. Imagine as a callus. Understood? And now if you want to open it, so you are using the broomstick to open it. You're putting that thing, you're using the broomstick, and as a result, the pores will be getting opened. Another, you can think that way also. If suppose you want to hold the water in the sink, and you right now there is no vegetable to wash it or what, so you will put 2 3 broomsticks to hold the water. Once you put the broomstick in between, obviously you are blocking the pipe only. So imagine that broomstick like a pea protein. Imagine that broomstick like a pea protein. So when you are putting a broomstick, obviously the water is not going down. So when I'm putting the pea protein, the food is not going down. As a result, leaf has to now fall. Is it clear? So pea proteins are helping in the callus pad formation also, and they help in dissolving it also at the time of spring season, so that the pore get open and the food can, matlab, phloem work can start. Okay, so I'm just writing it here. They help in sealing mechanism, sealing also, and it is also helping in conduction of food at the time of spring season, because that time they'll be opening the callus plug. Conduction of food. For opening, you can imagine you're using a broomstick to open the pores there. So this is an additional information with respect to your competitive point. Okay. Now, one more just I wanted to emphasize, right now in the sieve tube, if you see it is continuously tube, the food can come down easily. Okay. Food can come down through here, this pore, it can come down. If it is open, it can come down directly. It is the food is passing. But in case of the sieve cell, if you know sieve cell, the cell is individual maintained. So food conduction through sieve cell will be occurring in the zigzag manner. in the zigzag manner, just like in the xylem, the water was going through tracheids in the zigzag manner. Understood? Simple, you have to just remember that way only. Now let me explain you companion cells. Companion cells, very important line of NCERT. They are specialized parenchymatous cells. They are specialized. Their work is special, okay? They have given some special work, so that we call them, they are specialized parenchymatous cell. Again, it is very important line of NCERT. Please learn it properly. And I told you that they are coming from one cell only. One cell is dividing to form two cells. One is working as sieve tube element. Sorry, I should not draw nucleus, sorry. Another is working as a companion cell, okay, with a nucleus. So that is why sieve tube element and companion cells, they are coming from same mother cell. That is why they are known as sister cell. Okay, they are known as sister cell. And in between them, we have already discussed what they are present. They are having pit field connection. Pit field connections. They are present here. They are living cells and their nucleus controls the activity of sieve tube elements. Is it clear? In case of the gymnosperms, in gymnosperms, I told it that in place of the gymnosperms and pteridophyte, both actually, in gymnosperms and pteridophyte, they don't have companion cells. Instead of companion cells, they are having albuminous cells. And another name of albuminous cell is your Strasburger cell. Strasburger cells. Is it clear? This is again very important. Please learn it properly. It is known as Strasburger cell. I hope up till here topic is clear to you. The next component of the phloem is phloem fibers. And their fiber means they are made up of sclerenchyma. This you know very well. They are made up of sclerenchyma. Is it clear? And they are not found in primary phloem. They are not found in primary phloem. This is please important. They are not present in the primary phloem, but they are present only in the secondary phloem. And commercial fibers we are obtaining from the bast fiber also, just like a jute, flax, and all this, they are obtained from the phloem. One more component of the phloem is phloem parenchyma. This is the living component of the phloem. Okay. And it is the one which is actually storing the food. It stores the food. Now here it is important. It stores the food, that is starch. Obviously it will store starch, but other substance like resin, latex, and mucilage. And phloem parenchyma, very important line, they are absent in monocot. They are absent in monocot. Please learn it properly. Phloem parenchyma is absent in monocot and phloem fiber is absent in primary phloem. Is it clear? I hope up till here all your concept is getting clear. Now let me give you few additional information with respect to competitive point. Now see, these are the few additional information with respect to NEET point. So during the development of plant body, protophlloem get crushed. And the process by which it get crushed, that is known as obliteration. By the process of obliteration. Okay. Now main thing is this, what do you mean by the word obliteration then? So obliteration means after losing the protoplast, sieve elements are crushed and they are crushed by the adjoining cells and its content is absorbed by the adjoining cells. So this crushing and absorbing process of the sieve elements and companion cell is known as obliteration. Okay. So obliteration means after the loss of the protoplast, sieve elements is crushed by the pressure of adjoining cell and its content is absorbed by them. So this process of crushing and absorption of sieve tube elements plus companion cells by adjoining cell is known as obliteration. So this is the one thing which we'll be studying in the secondary growth, but I just wanted to introduce here so that you can understand the terms. So I hope up till here you are able to understand. So phloem topic is easy now. Thanks for watching.